Good afternoon and good morning, my dear colleagues, my dear delegates, wherever you are. A very warm welcome to this last session of our two-day conference. Today now for this session, we have two important and very distinguished speakers who will speak on two very critical areas of drug development. One is on wound healing approaches and the other one is on nanotechnology and nanomaterials in the drug development process. Dr. Joshua is currently working as a professor in the School of Science, University of Greenwich, United Kingdom. Dr. Boateng holds an MSc in pharmaceutical analysis from the University of Strathclyde. He undertook PhD and carried out research in pharmaceutical sciences under a Pfizer-sponsored project at the University of Strathclyde. He took up a postdoctoral position at the Nottingham Trent University. He joined the Faculty of Engineering and Science at the University of Greenwich in August 2007. He has more than 14 years of experience in academics. He is involved in supervising postdoctoral and PhD students for the various research projects. He is also a reviewer for many funding organizations' research projects. He has published more than 25 research papers in indexed journals. It's my pleasure to welcome Dr. Joshua Boateng for this session. Dr. Joshua Boateng, please take over and give your lecture.
Okay, good. Right, so I've already been introduced. So the title of my talk today is, my screen is vanished. That's it. It's advanced bioactive dressings for chronic wound healing. So let me tell you briefly a bit about the University of Greenwich. So we have 3 campuses. So our main campus is in central London in Greenwich, where the Greenwich Mean Time is located. I'm based in a campus called Medway in Kent, which is in the southeast. And then we have another campus in London in Avery Hill, in a place called Eltham. So a bit of an overview for my talk. I'll briefly give some background, and then I'm going to work through 3 case studies. The first one will be on medicated dressings, the second one medicated as well, and then I'll finish with honey and some work that we've been doing in this area. So if somebody asked, what is the point? 2 things really: chronic wounds, or let's say wounds in general. So for example, diabetic foot and leg ulcers are very difficult to treat. And actually, it can also kill people. There are several cases of amputations, very common with diabetic foot ulcers as well. And they cost millions to billions of pounds and dollars annually to various national health services, including the UK where I am. So in terms of background, wound healing is a very complex process. And the reason being, there are several biochemical, physiological processes and activities, and even cellular. And then there's also different stages of wound healing. So if you look at this cartoon here, when injury occurs, you have cells such as macrophages and neutrophils flooding the wounded area. You have platelets. So the platelets will try to stop the bleeding. The macrophages and neutrophils want to mop up infection and bacteria. That leads to hemostasis because fibrin is produced. And then all the other phases, inflammation, where the pain and swelling comes in. Proliferation, where you have loss of fibroblast cells produced to try and recover the wound, and eventually remodeling and scar formation occurs. And so typically we would try to treat wounds with dressings. In the past, we used traditional dressings like gauze, cotton, gels, and creams, but these are pretty useless really, because all they do is cover the wound and hope that the body would heal itself. Later on, you had what you call the modern dressings, or the moist wound dressings, such as films, foams, hydrogels, colloids, and wafers. And these typically create a moist environment that facilitates cell migration and eventual wound healing. But every dressing is expected to have particular characteristics, such as debridement and eventually protecting the wound from infection and contaminants. So these are the 2 common ones. What are some of the challenges then? So most dressings would only target one phase of wound healing. The 2 that I've described previously, they are biologically inactive, which means they do what you call passive healing. and then they depend on the normal body function to heal, because the body has an amazing means of healing itself. The other challenge is that no single dressing meets all the ideal characteristics that are showed over here. And so that creates an issue that needs to be addressed. So what are some of the solutions then? So you have what we call the advanced solutions. So you can use tissue engineered skin substitutes. These are typically used, as you can see in the pictures here, where there's significant tissue loss, and the same for the skin grafts. The problem with these two approaches is they're very expensive. Actually, in the case of skin graft, you have to create another wound, and actually that wound is very painful, and it's a form of surgery, so you need expert personnel to conduct it, and that makes it even more expensive and costly as well. And so there's third, fourth, and fifth advanced solutions, which is the interface where I work at the minute. You can use biological dressings where you're using biomaterials such as collagen and hyaluronic acid. So these are natural parts of the matrix. You can use medicated dressings. So this will be the modern dressings that I mentioned earlier, like the films and the hydrogels. You load them with various drugs that target different aspects of wound healing, like the pain and inflammation. You can load them with antibiotics to prevent or treat infection. Or if you want to be even more advanced, you can load them with growth factors, which actively will aid tissue or skin regeneration. And then these days, there's the natural products such as honey. The advantage of these three compared to the first two, which is the engineered substitutes and the graft, is these are readily available. And because of that, they are very cheap. So let me take you through our first case study. So in the first two, we work on three principles, which means that the dressings have to be biologically active. So they have to take an active part in wound healing. We want them to be multifunctional, so we use composite systems. So in this first one, we're using sodium alginate. Now that's a dressing on the market called sodium alginate, but we modify it with another one called polyox, which is just polyethylene oxide. So we're combining a natural bioactive polymer with a synthetic polymer, which is a very good bioadhesive. And then we load them with drug to make it multi-targeted. So here we loaded them with a module antibiotic, which is streptomycin for infection, and then we added diclofenac as an anti-inflammatory agent. But actually what we found was that this diclofenac actually enhanced the activity of the streptomycin, so we had synergistic activity. We produced them as wafers, so we get these nice porous structures, and this digital image actually looks like a foam or a sponge, but under the microscope is very highly porous, so it's very flexible, very easy to apply, and this porous nature enables it to absorb the exudate, and then it can hold on to the exudate as well, and then maintain the moist environment, which is one of the ideal characteristics required. We could also show that the drugs are both released. The streptomycin is more soluble, so you can see it's released more quickly as well, and to a higher percentage. But that's important because with microorganisms, you really want to hit them hard and kill a lot of them very quickly to avoid biofilm formation and developing resistance. And then the other one is released in a sustained way to deal with the pain and the swelling as well. And then, as you can see here, when you do the antimicrobial study in vitro, the zone of inhibition is very big for the drug-loaded wafer containing streptomycin and diclofenac. We compared it with 3 commercial dressings, all containing silver. And as you can see, there's a bit of zone of inhibition here, but the commercial dressings are not inhibiting as much as our drug-loaded formulation. And this is due to the synergistic activity produced by the diclofenac on the streptomycin. So the summary from the antibacterial study here was that our drug-loaded wafers are effective against the major wound infection-causing bacteria. There was a synergistic activity between the diclofenac and streptomycin, and we're able to inhibit both gram-positive and gram-negative bacteria. And this is important because most infections in wounds are mixed anyway, so you can't only target one particular organism. In fact, in some new work that we're currently doing, we're also targeting fungi and bacteria at the same time as well. And then this can reduce pain and swelling associated with chronic wounds. Now, this work is published in Journal of Pharmaceutical Sciences a while back, so if you're interested, then you're welcome to view it. So the second case study Here, we are trying to produce analgesic dressings for older adults, but instead of just rushing to make the dressing, we use what we call a stakeholder-based approach. So I kind of linked up with our department that has nurses who have a lot of experience dealing with patients from a sociological point of view as well. And so we go with the concept of acceptability, which is the ability of patients and their carers, including nurses and doctors, to use the product as is intended. And the principle is that to design effective dressings that patients would like, we need to ask them. So that's the basis of our study. And so we undertook an initial survey first with the nurses that treat the patients and with the patients themselves. And what we found was that pain is a serious problem and is poorly managed. And in fact, it's one of the reasons for poor compliance, because leg ulcers in particular are very painful. And we found that physical pain and the psychological trauma have a huge impact on the patient. In fact, we found that the patient anticipation of the pain was enough for them not to go to the clinic for treatment. And that the pain is highest during dressing change, which kind of makes sense. And that's why the moist wound dressings are important. You don't want to allow the dressing to dry out, because that is literally tearing up the newly formed skin tissue. And one of the things that the patients and the nurses told us was to avoid this constant regular changing of dressing. They would want a dressing that would have long-acting pain relief, for example, over 3 days. So based on these key findings, we set out to design our dressing. And this preliminary study is actually published as well. So again, you're welcome to investigate it further. And so we followed the initial principle from the first case study. We want the dressing to be biologically active, bioactive. We want it to be multifunctional. In this case, we're using hyaluronic acid, which is part of the natural skin matrix. So we're using a biomaterial again, another biomaterial. So unlike the first case study where we used natural and synthetic, here we're going for 2 natural biomaterials. And then we wanted it to be multi-targeted. So here, instead of diclofenac for the inflammation, we are using lidocaine because we want it to be treating acute pain. And then we use silver nanoparticles instead of antibiotics. So the summary findings was that, again, the dressing releases both the lidocaine and the silver. We were able to test the in vitro release of the lidocaine. For the silver, we proved the release by just doing the in vitro antimicrobial study. And it was able to inhibit the 3 bacteria we tested previously, E. coli, Pseudomonas, and Staphylococcus. They were very compatible from our MTT assay, so we knew that it would be safe for application. And then we did some in vitro wound healing scratch assay that showed that the cells, keratinocytes, and fibroblasts were migrating and closing up the wound on a glass slide. And again, we published this work as well in carbohydrate polymers. So I'm going to finish off with our third case study, and then hopefully we'll open up for some questions to allow some discussion. So we're going to talk about honey now. And I believe that every country has some form of honey, as long as there's bees in the country. And actually the use of honey is quite ancient, because as early as 2000 BC, there were medical writings for the use of honey for treating ulcers. And Aristotle also refers to its use for treating wounds. And recent interest in the use of honey is mainly because of antibiotic resistance. So really, there has to be alternative approaches. And it's quite funny that typically in pharmaceutical drug development, we develop new things from natural sources. We go the synthetic route, and then when we hit a roadblock like antibiotic resistance, then we go back to nature. So this is where we are. Now, this got so serious and scary that I remember a while back in around 2010, 11, 12, then Prime Minister David Cameron called an international conference because politicians were beginning to get worried that we might get to a place where a pandemic hits, not in the case of COVID viral, but in an epidemic where we have mass infections caused by bacteria and the antibiotics available cannot treat it. And so actually now there's an active political action between politicians, policymakers, and clinicians to try to address this issue. And so honey has quite a lot of bioactive components and properties that make it useful to treat wounds and treat infections. And actually, bacteria find it difficult to develop resistance because they have several avenues of attack. So they have antibacterial agents, you have the high sugar and low moisture, which bacteria struggle to survive in. And then, of course, it has acidic pH. So this is the bait that targets the infection. And then the bait that targets the wound healing itself, it has lots of antioxidants, it has natural proteins, it has minerals and phytochemicals. These are all together helping to boost wound healing. So there are a couple of products. You have Medi Honey, which is from Manuka. And then we have Sergi Honey as well. Just here, I just got a chat message here. Okay. So we published some initial work we did in this area where we tried to compare the antibacterial and functional properties of honey from Africa and Manuka honey for potential wound healing. So these are some of the key findings. So we're comparing Manuka, which is a gold standard, with two types of honey from Cameroon in Africa. And as you can see, the properties are comparable, and that tells me that we don't have to buy the expensive Manuka honey if we want to use honey in Africa, which saves a lot of money. So you can see here in the antibacterial study, the Cameroonian honeys, the CS and CW, had properties similar to the Manuka. And in fact, in some cases, when we've diluted it to 75% for Staphylococcus and Pseudomonas, it's actually doing better than the Manuka, which is quite interesting. And the reason why the dilution is actually enhancing the activity is because, honey is very viscous. So when you dilute it, then you enable the water-soluble components to be released and diffuse out more easily. So this was quite an interesting finding, which is unexpected. And in summary, Cameroonian honey exhibit properties that are relevant to wound healing. We were able to show that the antimicrobial activities were very good against common wound infection bacteria. And the good positive thing was that the activity was comparable to the current commercial standard, because if you went to a typical shop to buy Manuka honey in the UK, it's between 15 to 20 pounds. And here we bought these honeys in Africa on the market for just £1 or less. Okay, so you can see the difference. And we're also able to show that both the Manuka honey and the honey we got from Africa actually exhibited non-peroxide antimicrobial activity. So our key conclusions from the three studies so far is that a combined approach is required, which means we cannot use a one-size-fits-all, and we've got to target particular wounds using particular approaches. We can employ advanced multifunctional and medicated dressings using readily available materials to keep costs down, because it is really expensive. Anything that is chronic means that it's treated regularly and it can cost a lot of money. And then, of course, now we have the opportunity to exploit honey, which again is available on almost all continents and is relatively cheap. And I think it's particularly useful for poorer countries as well. So some ongoing work and future work. So currently we are formulating and testing dressings that incorporate these honeys. We're also testing a wider range of honeys sourced from different countries. And in fact, I've just had a project completed by some of my undergraduate project students where we tested honey from Canada, from London, and from Romania, where this project students was from. And hopefully we'll publish that soon. And in the future, we are hoping that we'll be able to do some preclinical animal in vivo work. And then once that is successful, then we would work with our clinical partners to undertake some clinical studies as well. And so with that, I want to acknowledge a few people. So Dr. Power, who was my former PhD student, he did all the work in case study 1. Dr. Video Catanzano is now moved to Italy. He was my postdoc who did the case study 2 in the actual dressing design. Keshu Dines did all the honey work, which we published. And I want to thank Samantha Lewis, who is our lead technician who helps with the microbiology studies. Professor Schofield and Dr. Dawkin, they led the study in the survey with our nurses and patients. to inform our second case study design. And I would like to thank the University of Greenwich for funding most of this work. And I just want to point you to, in fact, most of the work I've discussed here is actually published also in this new book, which was published last year. And you're invited to check it. And with that, I would like to thank you very much for your attention. And I'll be happy to take any questions.
Okay, thank you very much, Dr. Joshua, for your excellent presentation. Now we understand how important it is, this wound healing in the ultimate issue of patient care. If the wound healing is not properly addressed, if the correct strategy is not employed, we know it can lead to fatal results, and it can enormously raise the cost of the treatment also. And it is quite interesting also to know the various novel and new approaches that you are employing with various materials in designing novel wound dressing approaches.
Yeah.
I am also happy to note that you are actually collaborating with our university. Dr. Umar Sarheed is actually also carrying out a joint investigation with you, and you published a paper also.
Yeah, he's my good friend.
We look forward to more. Please go ahead.
No, I was saying that Dr. Sarheed is my very good friend. We collaborate and we publish a lot as well. So
we look forward to more active interaction with you in the years ahead.
Yes, absolutely.
But I have a couple of questions. I don't see many questions from the various delegates, but I can I ask you one or two?
No, please go ahead.
Yeah, you mentioned about various freeze-dried wafers as part of your novel approach to design the dressings. How do you control the porosity of the various dressings in the freeze-dried products?
Yeah, very good question. So first, you can do it in two ways. It's mainly through the freeze drying process itself or the type of materials we use. So with the freeze drying process, depending on the freezing rate, so if you freeze very slowly, then you get different what you call ice crystals. And it's the ice crystals that is determining the size and shape of the pore. So if you're able to manipulate the freezing rate to get the particular pore size you want, once the ice crystals evaporate, they leave that hole, and that hole is what forms the pore. Okay. So you can either freeze slowly or freeze very quickly, and then that's how you manipulate the pore size. But we also found that depending on the polymer, so for example, if you use sodium alginate, you tend to get elongated thin pores. And then if you use things like HPMC, they tend to give you nice circular shaped pores as well. So that's how we control our.
Okay, thank you. One more small question. What is this polyox, some material you are using?
Oh yeah, so it's a trade name. I should have used the proper name. It's actually polyethylene oxide.
Okay.
It's just polyethylene oxide. Again, a very cheap material to use. I normally use only cheap materials. Obviously, the whole point is that the cost should be affordable.
Thank you. Thank you very much. I don't see any other questions having been asked. So in the absence of any more questions, Dr. Joshua, thank you very much for your excellent talk. And we are really happy to note that you are associating with our faculty also. And we look forward to your collaboration in the future years. Thank you. Have a good day. Thank you.
Too.
Thank you so much.
Thank you. Bye.
Now we have the second talk by Dr. Mohammad Ezharul Hoque Chowdhury. He is currently working as a professor in Monash University. He has completed his graduate and postgraduate studies in biochemistry from University of Dhaka. He has worked as a researcher and group leader in the Tokyo Institute of Technology. He has got a lot of research work done in the areas of pharmaceutics, cell and molecular biology, pharmacology and toxicology, covering wide areas of science related to pharmacy. We are quite happy to note that, sir. He has published over 100 research papers in nanomedicine area, mainly as the senior author and as the first author. He has 7 published and pending international patents. He is also working as an editorial board member and reviewer for various popular and international indexed journals. Dr. Chowdhury will be speaking on another very important area of drug development, that is nanotechnology and novel nanomaterials. Dr. Chowdhury has a lot of expertise in the development of nanomaterials and in harnessing the nanotechnology to deliver drug molecules as a targeted drug delivery. So he will speak to us today on how this nanomedicine and nanotechnology is superior to the conventional materials and the drug delivery technologies. His talk is on pH sensitive biodegradable inorganic nanomaterials, a potential revolutionary delivery device for small molecule drugs and of course for macromolecular therapeutics. Very warm welcome, Dr. Chowdhury. Please take over and present your talk. Thank you.
Today I'm going to talk on pH-sensitive biodegradable inorganic nanomaterials, a potential revolutionary delivery device for small molecule and macromolecular therapeutics. So in my presentation, as part of the learning objectives, at first I'll try to explain the limitation of the classical drugs and the new generation macromolecular drugs, and how to overcome those limitations using application of nanomaterials. Secondly, I'll discuss the different types of nanoparticles and nanotherapeutics and their mechanism of action briefly. And finally, I'll discuss on the development of revolutionary nanotechnology platform based on pH-sensitive inorganic nanoparticles from our laboratory. Okay, let me discuss the background. What are the limitations and what are the mechanisms of the delivery of small molecule drugs? You know, most of the drugs we use still today are small molecule drugs, which means the drug size are below 1 nanometer. So quite small. Although we make different type of formulations and then we deliver in different routes, like oral route, the pulmonary route, intramuscular routes. But the size of the drugs are quite small, below 1 nanometer, as I say. So what happens if we deliver these small molecule drugs using different type of formulations with the help of different routes? So you know, the objective could be the local delivery or could be systemic delivery. So in most cases is a systemic delivery. So if we want to, like for example, if we want to deliver the paracetamol, the aspirin, and other small molecule drugs, you know, so we need to deliver to the blood. So using any of the routes. So using the oral route is the most famous. So after delivery, the small molecule drugs will come to the blood stream by any of the routes. And then in the blood stream, there are three factors: the blood flow, the plasma level of the drug concentrations, and the protein binding. You know, the drugs bind to the proteins in the plasma depending on their hydrophobicity, hydrophilicity. So these three factors control their traffic from the blood circulation to different organs. Okay, I'm not going to elaborate how they affect. So ultimately, the drugs will go to the different organs. Okay, and among the different organs, there could be target cells or non-target cells or organs. So then you can understand, using small molecule drugs with the help of any of the routes, the small molecule drugs distribute all over the bodies, basically, including target organs and non-target organs. Okay, the kidney is one of the targets. Kidney is one of the organs. So when the drugs goes from the blood to the kidney, they will be cleared because the size is very small. And when they go to other tissues, they will specific tissue or non-specific tissue, they will act, and they will finally come back to the blood circulation, and then they will be cleared by either by the renal excretion or by biliary excretion. So what I would like to show here, that small molecule drugs, they can distribute all over the bodies, and that's why we need to deliver huge amount of drugs in unnecessary manner. Number two, if the drugs are toxic, in fact, most of the drugs are toxic more or less. In case of chemotherapy drugs particularly, so you can imagine it will distribute all over the bodies and will kill not only tumor cells, but also normal cells, causing a lot of adverse effects. And that's why most of the drugs used clinically are mainly used just to increase the lifespan of the cancer patients, for example. Okay, and second and third is, if the drugs are hydrophobic, it's very difficult to deliver using even with the formulations. And they will have high affinity to the proteins. But if the drugs are bound to the proteins, actually they will not have any activity. They will not go into the cell. So they must be released. There is an equilibrium between free drug and protein-bound drugs. Only free drugs have activity and can go into the target cells or non-target cells. So this is the book I published in 2016. I think this is the first book, first extensive book elaborating on the nanotherapeutics that have been developed with mechanism of actions. So this is the cover photo showing that small molecule drugs are distributed all over the bodies. They are cleared by the kidney, here showing. But if we use nanoparticles, particularly multifunctional nanoparticles, so they prevent kidney clearance. They will go to the target cells, either by passive targeting or active targeting. They will go to target cells and will enter by endocytosis or will bind to the receptors. But if the nanoparticles are not multifunctional, are not hydrophilic on their surface, so they can be engulfed by macrophages of reticuloendothelial system. Okay, now this slide is showing how nanoparticles can alter the pharmacokinetics of small molecule drugs. Nanoparticles, so there is some misunderstanding. General people think that nanoparticles are very small. Actually, they are not smaller than small molecule drugs. They are much bigger than small molecule drugs. Nanoparticles are in the range of few nanometer to few hundred nanometers. On the other hand, small molecule drugs, as I mentioned, all are below one nanometers. So then how you can control the pharmacokinetics, the distribution of the small molecule drugs, because I told before that they distribute all over the bodies. So one is the green box showing. So with nanoparticles, we can increase the retention of the small molecule drugs in blood. That means we can increase the half-life of the small molecule drugs, because nanoparticles are bigger and they remain long time in the blood circulation. They cannot go all the tissues, and they can also, because with nanoparticle size becomes bigger, so they can also cannot be cleared by the kidney, because for the kidney clearance the size should be around 5 nanometer. So you can prevent kidney clearance, you can increase the half-life in the blood using nanoparticles. Second way is facilitating fast drug release. There are drugs like painkillers, for example. If you can deliver to the target cells quickly, so it can relieve the pain quickly. So you can make some formulation with nanoparticles for quick release of the small molecules drugs. You can also use nanoparticles to enable sustained release of drugs, especially, for example, diabetic drugs or even insulin. With nanoparticles, you can control so that glucose level, for example, gradually decline in the blood. Using iron oxide nanoparticles, you can these nanoparticles, as I'm showing, nanoparticles are acting as a carrier of small molecule drugs. So using magnetic nanoparticles, superoxide iron oxide nanoparticles, so you can deliver to the target cells with the help of an external magnet, because iron oxides will be attracted with the external magnet, and the iron oxide nanoparticle will carry the drugs. So for the hydrophobic drugs, showing by the yellow box, hydrophobic drugs, most of the drugs developed in the pharmaceutical industries are hydrophobic, and they are discarded, because so hydrophobic. But using nanoparticles, even previously discussed, this discarded drug can be reused, because it can help to increase the solubility of the drugs. And also nanoparticles can be used for the passive targeting, especially for the tumors. The tumor has a leaky vasculature, pore size of around few hundred nanometers. So if you can design your nanoparticles of few hundred nanometers, so they will be selectively go into the tumors. They can pass, extravasate the tumor vessels and enter into the tumor environment. And at the same time, tumor has underdeveloped lymphatic system. So that's why the release drugs can retain in the tumor for a long time. This is what we call EPR effect, enhanced permeability and retention effect. And finally, you can even coat the nanoparticle surface with some ligand, like transferrin, because some cells, target cells have receptors for transferrin, like folic acid receptor overexpressed in many cells. Even antibody, you can use antibody as a ligand, because many cancer cells or other targets have the antigen receptors that can recognize the specific antibodies or antibody fragments. So this is part of active targeting. So now you can imagine that how nanoparticles, nanotechnology can revolutionize the medicine. Hopefully, in the next few decades, you will see a huge difference of the medicine. In fact, there are many nanomedicine products already on the market, and this approval rate will be increased day by day. So now let's have a look what type of macromolecular drugs are present or could be developed. The green one showing protein-based macromolecular drugs. And that can be further categorized into three groups. One is protein as independent therapeutic drugs. In the market now, so many therapeutic proteins, including insulin, many monoclonal antibodies, many enzymes. So they are independent therapeutic drugs. Protein can be as a drug carriers like albumin. Even in the market you will find albumin as a carrier of drugs, anti-cancer drugs. Even protein can be delivered using nanoparticles, because, like for example, monoclonal antibody or monoclonal antibody fragments or enzyme, they have short half-life. But with the help of nanoparticles, you can increase the half-life in blood. And in that way, you can minimize the cost and also side effects. Another application is DNA-based macromolecular drugs. It's a part of gene therapy. The many genetic disease and acquired disease, those are caused by the mutation or dysfunctions of the gene. So in that case, like even for cancer, in cancer, there are important genes like p53 and other tumor suppressor genes are mutated. So if you can deliver the genes like tumor suppressors, so it can be a treatment option. But for this, For this, you need a nanocarrier because the nucleic acid are very sensitive to degradation by nucleus. Another is oligonucleotide, antisense oligonucleotides, or siRNA, small interfering RNA. So they have opposite effect like gene. So like, for example, in cancer, cancer are also caused by mutation in proto-oncogenes, converting them into oncogenes. So they are harmful genes. They cause unlimited division of cancer cells. So if you can deliver antisense oligonucleotides or small interfering RNA using nanoparticles, because they are also sensitive to nucleus, so you can block the function, you can block the production of those proteins. So that is also very huge area of application, and there are many clinical trials going on. You can use nanoparticles for vaccine, like for COVID-19 vaccine. All vaccines in the market are mRNA based, for example, or others, DNA based or protein based. For the mRNA based or DNA based, you must need a carrier because mRNA are even more sensitive than DNA or siRNA. So, like for example, in case of Moderna, they are using lipid based particles. Some are using viral vectors. So you need a carrier at least. So that is also a very important area for mRNA, for vaccine development. You can also deliver using nanoparticles the imaging essence. Imaging is essence, like even it could be fluorescence or other contrasting agent. At the same time, drugs. So it is part of theranostics. So you can deliver both imaging, you can do diagnosis, detection of your delivery system, as well as you can deliver drugs. Finally, macromolecular or nanoparticles can be used for the product therapy, and it has two types. One is you can deliver, at first you can deliver the enzyme tag antibodies, for example. The enzyme can cleave the product into active drugs. At first you need to deliver, so they will bind to the target cells using antibodies, and then you need to deliver product. And product, even though they distribute all over the body, so only the drug that reach the target site that has the antibody directed enzyme, that in that case the drug will be clipped in that particular site and will be active. Another way, you can use nanoparticles to deliver the genes of that enzyme that will break down the product, so using nanoparticles in a targeted manner. So now it also shows that macromolecular drugs or nanoparticles or nanotherapies have huge application. So there are different types of nanoparticles: liposomes, dendrimers, solid lipid-based nanoparticles, even viral particles, even though they have some potential adverse effect, carcinogenicity, immunogenicity, polymeric micelles, gold nanoparticles, polyplex, and the pH-sensitive nanoparticle that we have developed. This is an important nanotechnology platform that we pioneered, and it has the property that is absent in other existing nanoparticles. This is pH sensitive in the sense that when they go to the target site, it could be acidic tumor environment or intercellular acidic endosomes. So these particles will be dissolved quickly. And this is how they can release the drugs. The drug could be small molecule drugs or macromolecular drugs like genes, like siRNA, like proteins. And it can also help endosomal escape for macromolecular drugs. If I have time, I will explain. So this is a nanotechnology platform we developed. I will explain later. So these are the fascinating properties of our pH-sensitive inorganic nanoparticles that I developed when I was doing my PhD, in fact, and then I extended this field, and I have and my team has developed other type of inorganic nanoparticles that was overlooked so far, even though they are the simplistic nanoparticles, but they could not receive the attention of the researchers. The researcher focused liposome, polymeric nanoparticles are inorganic, non-biodegradable particles, but they have not focused this area. So why they are excellent candidates? Because these particles, even though this is 100 nanometer particles, So this consists of very small tiny particles, like 5 nanometers, for example. And these particles has positive and negative charge because these are consist of small particles that are present, in most cases, present in our bodies. So they form the crystals, and they have positive and negative charge on their surface. As a result, they can bind drugs, DNA, proteins by ionic interactions. All are ionic interactions. The particles form the crystal by ionic interaction. They bind drugs, ionic interaction. Even when you modify particle surface with some ligand or other hydrophilic, also by ionic interactions. Not any covalent interaction. And that's why they are very sensible at acidic pH or other conditions to be degraded and to release, unlike the covalent interactions, which is very strong. And you can also control the size of the particles. That is important for passive targeting, as I said, the tumor vasculature, for example, leaky. So you can do passive targeting by controlling the size of the nanoparticles. And the most important property, as I mentioned, they are pH sensitive. So when they go to the acidic environment, into the target cell or target site, there is complete dissolution, causing drug release. This is the most fascinating property, and that's why, because our particle has these properties, that's why we need very small amount of drugs to get a therapeutic effect in animal models. At the same time, our system also has less side effects, because when they dissolve, they release the salt, and the salt component can be processed by our body system, because salts are present in most cases in our body, and that's why cells have developed the mechanism to transport out from the cells. So this slide is showing. Just give you one example of our particle systems. It's showing that the particle basically consists of 3 salts, three ions: calcium, phosphate, and carbonate. Carbonate that is present in soda, in bicarbonate drinks. That is the essential component for these particles. And then with addition of 2 more salts, for example, iron, magnesium, for example, so you will have 5 ions, 5 salts to make one multi-cation, multi-anion particles. Similarly, you can replace iron, magnesium with strontium, magnesium, for example. So that will also have 5 salt-based nanoparticles. And why we are changing? To get the diversity, because diversity is very important. To, depending on the drugs, depending on the target site, you need a diversity. You need to design, and diversity helps drug binding. It helps protein corona formation. That is very important. What is protein corona? So when you inject nanoparticles, blood has thousands of proteins. They will immediately coat your particle surface. That is called protein corona. And because of this protein corona, the pharmacokinetics of nanoparticles are very different depending on the protein corona type. So if you have the diversity with different type of salts, for example, so your protein corona will be different. If you can control the protein corona, your pharmacokinetics will be different. You can target to different organs as well. Okay, finally, it will be degraded after going to target site, because all nanoparticles we develop are pH sensitive in order to release the drugs in the target site. So this is another way. So you can make the nanoparticles with different salts with our formula, but to control the protein corona or to increase non-specific interaction with proteins, to reduce the off-target distribution, you can modify the surface with some biomolecules, organic molecules. For example, we recently succeeded for the first time Krebs cycle intermediates like citrate, succinate, alpha-ketoglutarate. They have the carboxyl group they can easily bind on the surface because all are ionic interaction, as I told. Another is biotin PEG. PEG cannot interact, but if you have biotin, biotin has some ionic group, they can bind on the surface of the nanoparticles. So ultimately, your particles will be pegylated, PEG-coated particles. So that will give hydrophilic surface. So, and that is why we can prevent intake by the heart muscle. We can prevent intake by the brain cells by using this hydrophilic coating. Okay, and also we can control the size, shape, and surface charge. And also we can increase the tumor accumulation by this way. So this slide is showing. So if you want to carry genetic materials like plasmid DNA that carries genes of interest, or small interfering RNA, or antisense oligonucleotide, or miRNA, or others. So what will happen? It will bind by ion interactions, and then it will enter by endocytosis, and endosome has proton pump, and these particles are pH sensitive, so they will accept the proton, hydrogen ion, and will dissolve quickly. And this dissolution will help release of the drugs, that means DNA or siRNA or other drugs. And this also helps because this dissolution, release lot of ions, and these ions could create osmotic pressure, and so water can enter and can lead to soiling of the endosomes and rupturing. So this is the hypothesis, and I think that is why the DNA siRNA can efficiently escape, and that is why we need very small amount of genes and siRNA to get a therapeutic effect. So these are giving some examples, our successful studies. So this study was done using the carbon apatite nanoparticles, our original nanoparticles. So what we did, we delivered cyclophosphamide anti-cancer drugs in a syngeneic mouse model of breast cancer. And you can see this is just control showing no effect of the nanoparticles on the tumor regression, is so that no side effect, no body weight change. But when you deliver the cyclophosphamide with your nanoparticles, the tumor size, as represented by y-axis, is going down. So there is no further growth, inhibiting the tumor growth, tumor regression by cyclophosphamide coated, embedded nanoparticles compared to free drugs. And this slide showing that the tumor becomes very small with our nanoparticle loaded cyclophosphamide. So what we did, because it's a product, so we delivered our nanoparticle to the liver, because liver vessels also leaky, like tumor. It's more leaky, in fact. So you can deliver nanoparticles, even bigger nanoparticles, and tumor will receive and will activate the product into active, because liver has the enzyme to activate. So that's why. So we at first we deliver the product to the liver using nanoparticles. Liver activate the drugs, distribute to the others, including the tumor, including the cancer cells. So this is another interesting study we did. We delivered small interfering RNA targeting different type of growth factor receptors. For your information, for the cancer development, the growth factors are the prominent causes. Growth factors are expressed by oncogenes because of mutations from proto-oncogenes. And these growth factors are present on the cell membrane, and they receive the signal when they becomes activated or overexpressed. They receive the signal from the ligand continuously, and that's why cell division is continuous, and that's causes tumor, unlimited cell growth. So that's why if you can block the formation of the growth factor receptors, that is very important way of controlling, of killing the cancer cells. And also another one is BCL2, is anti-apoptotic genes. So this also is oncogenes, important oncogenes. So in this study, we deliver growth factors, siRNA, targeting like EGFR, HER2, IGF-1, estrogen receptors, and BCL2. Around five, and their combinations. And you can see from this Western blot that with siRNA targeting the growth factor receptor like estrogen receptor, HER2 and others, the MAP kinase and AKT, because this AKT pathway and MAP kinase pathways are two different pathways. They are linked to each other. They are responsible for tumor cell proliferation, division, and survival. So you can see that with siRNA delivery using nanoparticles, so the activation of this pathway is down-regulated clearly. And when you combine, the effect is more drastic. Because of time limits, I cannot explain everything here. So, like for example, we got estrogen receptors, and like here is showing three growth factor receptors. The down one, the tumor regression is more prominent with the siRNA targeting estrogen receptors, HER2 receptors, and also EGFR receptors. Three receptors delivered in a combination way, and that's why there is a synergistic effect of tumor regressions, and also is shown by the deactivation of the pathways with this combination. Similarly, we succeeded with BCL2 and estrogen receptors, but I'm not showing here. So this is another slide that K.V.R.N.S intermediate, like alpha-ketoglutarate modified nanoparticles, can drastically reduce the tumor size, as shown by the red color at the down. No tumor growth at all. When we deliver cyclophosphamide using alpha-ketoglutarate modified, that is one of the K.V.R.N intermediates present in our body. Okay, and why is decrease? The decrease is because it increased the half-life. This is showing the plasma circulation of the doxorubicin. So after intravenous delivery, we measure the plasma half-life, and you can see 2 hours and 24 hours with our nanoparticles, alpha-ketoglutarate modified nanoparticles. It almost 10 times increase in the blood level of doxorubicin, both in 2 hours and 24 hours. So definitely it increases plasma half-life, and that's why it gradually delivered to the tumor. It prevents renal clearance. It prevents distribution to other non-specific organs. And most interesting is that we also check the distributions, that with our alpha-ketoglutarate modified or succinate modified, there is huge tumor accumulation compared to free drugs, only naked particles, only particles with cyclophosphamide. And interestingly, you can see the heart. In case of heart, there is a drastic reduction of accumulation of doxorubicin for our nanoparticles, especially K.V.R.N intermediate modified particles. And you know, doxorubicin is an important drug, commonly used. Even I had a friend who had cancer who finally passed away. He also received this drug, but this drug was so active for his cancer, but the drug was discontinued after few cycles, like 6 cycles, because it is recommended that it may cause cardiotoxicity, this doxorubicin, so should be discontinued. But using nanoparticles, you can see that it can prevent heart muscle uptake, so paving a new area of exploration in the clinical trials. So this I have time limitation, so I will go a bit faster. So this slide showing that we also use the pegylated nanoparticles like biotin PEG I told. So we coated by ionic interaction because biotin has some ions, some charges. So, and this is how what we succeed by using biodistribution. We are showing that we use here different 2 types of nanoparticles, iron, magnesium, carbon apatite, and only carbon apatite particles. And we compared with our PEG. It shows that with the pegylation, tumor accumulation increase, and also it reduces the brain uptake, and also uptake by the heart. And this slide showing the gemcitabine. Gemcitabine also was encapsulated into our nanoparticles, into our pegylated nanoparticles, and you can see that the tumor regression with our pegylated carbon apatite nanoparticles, the tumor regression is so significant compared to only carbon apatite gemcitabine. and also compared to free gemcitabine. And also we check the protein corona by LCMS. So this is unmodified carbon-reported particles and modified. You can see the pegylated particles bind less amount of proteins. It's mainly transport proteins. So so that means by modifying PEG, so you can make it more selective. You can prevent off-target distribution. And that's why when you check the biodistribution, there is more tumor accumulation with our pegylated nanoparticles. There is less off-target effect with our pegylated particles like liver, spleen. So this is my last slide. So in this study, we collaborated with a laboratory in Germany. So they wanted our nanoparticles because I told my collaborators that these nanoparticles are so simplistic, and you can incorporate antibodies just by mixing, by ionic interactions, without losing the activity of the proteins, loaded proteins. So that's why they were interested. I just sent by DHL, and they make the nanoparticles in presence of antibody fragments, FITC antibody, specific to this antibody fragment was specific to some antigens. After making this formulation, okay, they treat it with the tissues, human tissues. That is shown here. This slide is showing that they treated squamous epithelium and also Barrett esophagus. Barrett's esophagus is part of the Barrett's dysplasia. Is the private state of the cancer. So you can see, compared to free antibodies, this is giving lower signal. Our antibody coated, nanoparticles giving stronger signal compared to the free antibodies in both squamous epithelium as well as Barrett esophagus. So opening the area of using these nanoparticles for molecular diagnosis. So this is another big area that we can explore actually. So this slide is showing that there are many more applications of the nanoparticles, especially this type of nanoparticles, which are biocompatible because they are biodegradable, and they can release the drugs. So one is, as I mentioned, targeted delivery of the drug, different type of drugs, as I mentioned. It can also help to chemosensitize using siRNA delivery. So we have published some papers that with some specific gene silencing, gene knockdown, we can sensitize cancer cells to specific drugs. We can deliver proteins or antibody fragments. Like currently the therapeutic proteins act on the membrane, but there are many targets into the cell. So using nanoparticles, you can also deliver the therapeutic antibodies or protein into the cell because they will go by endocytosis. You can deliver DNA protein, as I mentioned, for vaccine delivery is another area that we can use the nanoparticles for mRNA delivery, for example, or DNA delivery for vaccine. We can deliver therapeutic imaging as part of theranostic I mentioned before, and also confocal microscopy-based endoscopy that I collaborated with the German lab, and also can be used to extract, to purify proteins and other molecules using nanoparticles. So I would like to acknowledge all my lab members. I'm sorry I could not show here the pictures of my lab members and my collaborators, and thank you very much all for your kind attention. Thank you very much.
Thank you very much, Dr. Chowdhury, for your very lengthy and detailed talk on how nanotechnology and nanomaterials can really do wonders in delivering small drug molecules, particularly the anti-cancer drugs. You also have spoken about the various pharmacokinetic issues that are very critical in designing a very successful delivery module for a nanoparticle system. I also want to know from my side, there are a few questions, but I just would like to ask, can I ask one or two questions?
Yes, please.
Yeah, did I hear you correctly whether the doxorubicin pharmacokinetic changes that you brought in with this nanoparticle technology is 10 times more than what it is
10
in
times
one
more than
patient.
fluid drugs.
Yeah, exactly. You could increase by 10 times, right? That's what I
Yes.
understood. So we can bring such a large change in the pharmacokinetics of a drug also, in addition to various other therapeutic benefits, right?
Yes, right.
Okay, good. After listening to your seminar, I'm sure, Dr. Chowdhury, many young researchers will be definitely motivated